In this section, we'll be discussing enzymology. Now, in order to understand enzymology, we've got to understand something about energy and how the body uses energy. And this will be important in the rest of the lectures that we do for biochemistry. Cells have to have energy in order to perform the tasks they need to function properly. Things like the synthesis of macromolecules, for example, or the transport of molecules across membranes, or movement of cells, for example, all require energy. Now, cells obtain this energy by doing particular chemical reactions. And the amount of energy that's consumed or liberated during a particular chemical reaction is defined as delta G, or change in free energy. Now, the delta G value for any reaction indicates whether or not that reaction is spontaneous. That is, the probability that it can proceed without putting any energy input into it, without providing energy from the outside. You know, can this reaction proceed or not? Now, that causes us to categorize reactions in two different ways. One is an exergonic reaction, in which the delta G, or free energy, change in free energy, is negative. When the change in free energy is negative, that means that the reaction can proceed spontaneously. This is an energy-producing reaction. An endergonic reaction, on the other hand, is one in which the delta G is positive. In other words, an input of free energy is required to drive that reaction. This is an energy-consuming reaction. Now, this is illustrated in the following schematic diagrams. Now, for a reaction that proceeds from A to B, let's say A is a substrate and B is a product. On the left-hand side, in an exergonic reaction, notice that it proceeds from a high-energy state, G, in A, to a low-energy state in B. And so the delta G in this case is going to be less than 0. If you subtract A from B, you get a negative number. And that means that there's been a release of free energy. This is an energy-producing or an exergonic reaction. On the other hand, in the schematic on the right, we show an endergonic reaction. In this case, you start with a substrate at a low energy level and produce a product that has a higher energy state. So that means that there has to be an input of energy to drive the substrate, the low energy substrate, to the higher energy product. And so again, if you subtract energy of A from the energy of B, you're going to end up with a positive number. Delta G will be greater than 0, which means we have an energy-consuming or endergonic reaction. Now, a lot of the biosynthetic reactions in a cell are endergonic. They require energy. For example, you're making things that are more complex. You're synthesizing things, creating things, and all of this requires energy. Now, the cell drives these energy-requiring reactions by coupling them with exergonic reactions, which release free energy. For example, if you're going to form a peptide bond between two amino acids, it requires an input of energy. It's an endergonic reaction. This energy is provided by coupling the reaction with a hydrolysis of ATP, which is an exergonic reaction. So you can see in the sum that the reaction combining amino acid 1 with amino acid 2 to form a dipeptide has a delta G that's greater than 0. It's an endergonic reaction. But the hydrolysis of ATP to ADP plus inorganic phosphate is an exergonic reaction where the delta G is much less than 0. So if you add these things together, amino acid 1 plus amino acid 2 plus ATP can cause the formation of a dipeptide bond with the release of ADP plus inorganic phosphate. The net result is a reaction where the delta G is less than 0. So this reaction can proceed spontaneously then because it is a net exergonic reaction when you combine the two reactions together. Now, delta G tells us about the direction of a reaction. Do we move from product A to product B, or do we move from substrate B to product A? Which direction is the reaction going to go in? But most times in the conversion of A substrate to B product, there's some transition state, which we call A-B, or A-B, that's an intermediate form between the substrate and the product. The formation of this intermediate usually requires more energy than exists in the substrate itself. And so it's a higher energy state. So in order to allow the reaction to proceed, you have to have an input of energy to overcome this barrier. The degree of this barrier is called the energy of activation, as shown. The greater the energy of activation, the slower the reaction, and vice versa, because the greater input of energy is required to get the reaction started. Now, there are certain things called catalysts. Catalysts are able to lower the energy of activation, and this means that there's less energy required to start the reaction, and so the rate of the reaction increases. In biologic systems, the catalysts that are used are called enzymes. These are usually proteins which decrease the energy of activation and cause reactions to proceed at a faster rate. Now we can categorize the enzymes in biologic systems into 6 different classes, as listed here. The first of these classes is what we call oxidoreductases. These are enzymes which catalyze oxidation reduction reactions, in which electrons are transferred from one molecule to another. And they have names that usually end in things like dehydrogenase or reductase, oxidase, peroxidase, catalase, hydroxylase, and so on. That you can, if you recognize those names as oxidoreductases, you know that these are enzymes that are involved in redox reactions, oxidation reduction reactions. For example, alcohol dehydrogenase is an oxidoreductase. It catalyzes the oxidation of ethanol to acetaldehyde using NAD+ as a cofactor. And as ethanol is oxidized then, it loses its electrons and donates them to NAD+. NAD+ is reduced to form NADH. So this is an example of a redox reaction catalyzed by an oxidoreductase. Transferase enzymes are enzymes which catalyze the transfer of some functional group from a donor molecule to an acceptor molecule. And these are things like transaldolases or transketalases, acyl, methyl, glucosyl, phosphoryl transferases, and phosphomutases, for example. An example of this is a protein kinase, which transfers a phosphate group from ATP to a serine or threonine residue in some protein. So we've moved a functional group from a donor to an acceptor, and that's the function of a transferase enzyme. Hydrolases break things up. They cleave off functional groups and usually transfer them to water. These are things that have names like esterase, glycosidase, peptidase, phosphatase, and so on. The general reaction is the hydrolytic cleavage of particular bonds, for example, a carbon-oxygen bond, a carbon-nitrogen bond, a carbon-sulfur bond, or an oxygen-phosphorus bond. For example, phosphatases remove phosphate groups on serine and threonine groups and combine them with the hydroxyl group from water. So they're cleaving off a functional group. So that's why they're defined as hydrolases. Lyases add or remove water, ammonia, or carbon dioxide from macromolecules. And these have terms like, or names like decarboxylase, or aldolase, or hydratase, dehydratase, so on and so forth. An example of this is pyruvate decarboxylase, which catalyzes the conversion of pyruvate to acetaldehyde, and in the process releases a molecule of CO2. So because it produces CO2, we call it a lyase. Isomerases don't add or remove anything from a molecule. Rather, isomerases simply change the configuration of that molecule by the intramolecular transfer of functional groups. The molecular formula of the compound stays the same. These things have names like isomerase, or racemase, or aperamase, or mutase. An example of that is phosphoglucose isomerase, which converts glucose 6-phosphate to fructose 6-phosphate. Now, glucose 6-phosphate and fructose 6-phosphate have the same chemical formula, the same molecular formula, but you can see in the schematic at the right they have a different molecular configuration. Another type of enzyme is the ligases. These enzymes catalyze synthetic reactions where two molecules are joined together. And this usually occurs at the expense of a high-energy phosphate bond from ATP or some other nucleotide triphosphate. These have names like polymerase, synthetase, ligase, and carboxylase. Some of these we've encountered already in talking about DNA and so forth. For example, DNA polymerase is an enzyme which joins nucleotides together, and so we would call it a ligase. Now, these are the 6 different types of enzymes. There's a couple other definitions that you should be aware of when we talk about enzymes. Just be familiar with these terms. For example, a cofactor. A cofactor is a small organic or inorganic molecule that certain enzymes require for activity. For example, there's an enzyme called lysine oxidase, and lysine oxidase has to be bound to a molecule of an ion of copper in order for it to function properly. So copper ion, in that case, is a cofactor. Prosthetic groups are molecules that function as cofactors, but they're also tightly bound to the enzyme. For example, a hemoglobin molecule contains a heme prosthetic group, and that is a cofactor necessary for the functioning of that molecule. We call those prosthetic groups. Now, whether or not an enzyme has its cofactor or prosthetic group determines the name that we use for it. An apoenzyme is the enzyme form that lacks its cofactor or prosthetic group. This is usually inactive because it doesn't have the cofactor or prosthetic group that's necessary for its functioning. A holoenzyme, on the other hand, is the complete enzyme that carries its cofactor or prosthetic group and is now active. That's called a holoenzyme. Now, when we consider enzymes and how they function, one of the things that we talk about is kinetics. Now, kinetics is important because kinetics describes the rates of reactions, how fast reactions occur, and that's very important in biological systems. Consider the following reaction where we have an enzyme labeled E, some substrate, which we call S, ES, which is the transitional state, the complex between the enzyme and the substrate, and then the enzyme plus some product. So in this chemical formula, we have an enzyme combines with its substrate to form an intermediate state, and then that substrate is converted to product with release of the enzyme. Now, each of these steps has a rate constant. The rate constant, which is called K, expresses the activity of the enzyme, the number of substrate molecules converted to product in a given time by a given number of enzyme molecules when the enzyme is saturated with substrate. So this is the rate at which each of these reactions may occur. So the rate at which the enzyme combines with substrate to form the transition state is called K1. That reaction is usually reversible, so the rate at which the enzyme dissociates from its substrate is called K2. And then the conversion of the enzyme substrate transitional state to enzyme plus product is an irreversible reaction, the rate of which is defined by K3. In most situations, the rate of enzyme-catalyzed reactions is dependent upon the concentration of the substrate. And how the rate and substrate concentration are related to each other depends on what we call the order of the reaction. In what we call a first-order reaction, the rate depends upon the concentration of a single substrate. There's only one substrate whose concentration defines the rate at which the reaction is going to proceed. In a second-order reaction, the rate depends on the concentration of 2 different substrates. And so, depending upon the varying concentrations of those 2 substrates, you'll get a different rate of the reaction. Now, when we saturate a system with substrate, that is, we increase the substrate so much that further increases do nothing to the rate of the enzyme. When we do that, then the rate no longer depends upon the substrate concentration. You can keep increasing the substrate concentration forever, and the rate will never change. This is called zero-order kinetics. Now, the relationship of substrate concentration to the rate of the reaction is defined by a particular equation that we call the Michaelis-Menten equation. Now, this is one of those other equations that you really need to memorize to answer the questions on the USMLE. So you need to memorize this reaction. This equation is V, which represents the rate or velocity of the reaction, is equal to V max, which is the maximal velocity of the reaction. The velocity of the reaction when the substrate concentration is saturating, times the substrate concentration divided by the substrate concentration plus a term that we call Km. Now, Km is the Michaelis constant. This is defined as the substrate concentration when the velocity is half maximal. So if Vm is 10, for example, Km would be the substrate concentration that causes the rate to be 5. Now, the relationship of substrate concentration to the rate of the reaction is defined by a particular equation that we call the Michaelis-Menten equation. Now, this is one of those other equations that you really need to memorize to answer the questions on the USMLE. So you need to memorize this reaction. This equation is V, which represents the rate or velocity of the reaction, is equal to V max, which is the maximal velocity of the reaction. the velocity of the reaction when the substrate concentration is saturating, times the substrate concentration divided by the substrate concentration plus a term that we call Km. Now Km is the Michaelis constant. This is defined as the substrate concentration when the velocity is half maximal. So if Vm is 10, for example, Km would be the substrate concentration that causes the rate to be 5. Okay? Now, in this equation there are two variables and there are two constants. For a given enzyme, in a given situation, the Vmax and the Km are constant. They don't change. Okay? The velocity, the rate may change. In fact, that's the variable that we're interested in. And the substrate concentration will change. And then this equation then describes the way that the rate changes as the substrate concentration is either increased or decreased. Now we can graph this. We can graph velocity or rate versus the substrate concentration. And when we do that, the graph looks like this. It's a rectangular hyperbola. And so you can see that when substrate concentrations are very low, as you increase the substrate concentration, the velocity increases rapidly. As the substrate concentration continues to increase, the rate at which the velocity increases levels off until it reaches a Vmax, the point at which no matter how much more you increase the substrate concentration, the rate will not change. We've also defined for you here the Km. Now in this particular case, the Vmax is 20, so the Km is going to be the substrate concentration at which the velocity is 10, the rate is 10. And so if you graph that, if you find the point on the graph at which the rate is 10 and trace that down to the substrate concentration, you'll get the Km, which in this case is 5 millimolar. Now, if we want to determine these parameters experimentally, you measure the rate of the reaction at various substrate concentrations, you plot them on a graph, and you derive the equation. But the equation of a hyperbola is complicated, and so what we do is we simplify it by rewriting the equation as a double reciprocal. That is, we take both sides of the equation and flip them over. And this is called the Lineweaver-Burke equation. So when you flip it over, you get 1 over the rate, or V, is equal to Km over Vmax times 1 over the substrate concentration plus 1 over Vmax. Now what you'll notice about this is the equation for a straight line. You may be familiar with this equation from algebra as y equals mx plus b. Well, in this particular term, the y is 1 over v, the m is Km over Vmax, the x is 1 over the concentration of the substrate concentration, and the b is 1 over Vmax. So if you plot out the rate at different substrate concentrations, what you get is a straight line, as shown in this particular graph here. Now this is important then because you can use this graph then to figure out what Km and Vmax are. If you have a graph like this, the slope of the line is going to be Km over Vmax. And the y-intercept is going to be 1 over Vmax. So if you know what the slope and the y-intercept are then you can figure out what the Km and Vmax will be. And then you can calculate what the rate will be at any substrate concentration. So here's a question that uses these concepts. A particular enzyme has a Km of 20 micromolar. When the substrate concentration is 10 micromolar, the reaction velocity is 500 micromoles per minute. What would the reaction velocity be if the substrate concentration were doubled? Okay? Is it A, 250 micromoles per minute, B, 750 micromoles per minute, C, 1,000 micromoles per minute, or D, 1,500 micromoles per minute? I'll give you a minute to calculate that, and then we'll look at the answer. The answer is B. All right, let's analyze this question. To figure out, first of all, we need to figure out what it is that the question wants us to find out. The question wants to know what the reaction velocity will be at a given substrate concentration. Okay? So in order to know what the velocity will be at a given substrate concentration, you have to know what Km and Vmax are. Okay? Well, we already know what Km is. Km is given to us as 20 micromoles, so we need to find out what Vmax is. And we can do that by plugging in some numbers into the equation. So we know that Km is 20 micromoles, and in the beginning of the question, they tell you that when the substrate concentration is 10 micromoles, that that the velocity is 500 micromoles per minute. So you just plug in the substrate concentration and the rate with the Km that we already know, and then you solve for Vmax as shown in this slide. If you solve for Vmax, what that tells you is Vmax is 1500 micromoles per minute. Okay? So now you know Km and you know Vmax. So what you want to know then is what is the rate going to be if you double the substrate concentration from 10 micromoles to 20 micromolar. So in this case, because you know the Vmax and you know the Km, you can simply plug things in. So V equals 1500 then times the substrate concentration, which is 20, divided by the substrate concentration 20 plus the Km, which is 20. And if you solve for that, you get 750 micromoles per minute. Now, I hope you got that right by calculating it exactly as I've described. But I want you to realize that there's an easier way to solve this question. If an enzyme has a Km of 20 micromolar, then you know that the velocity is automatically going to be half of Vmax at that point. So if you solve for Vmax, the velocity at the Km of 20 is always going to be half of Vmax. Well, when we double the given substrate concentration, 10 micromolar, what we get is 20 micromolar, which is the Km. So since we know that at the substrate concentration equal to Km is half of the Vmax, then all we have to do is take the Vmax and divide it by 2. 1500 divided by 2 is 750. And that gives you sort of a shortcut to the answer for that question. Now we've been talking about rates and how enzymes control rates of chemical reactions. Now you can imagine that if you want your body to function properly, if you want it to be adaptable to different situations, you want to be able to regulate the rates of various reactions. And so this is accomplished by regulating the activity of the enzymes which catalyze those reactions. Now there's a lot of different ways that enzymes can be regulated, but some of these are inhibition, allosteric regulation, covalent modification, protein-protein interactions, and you can also regulate it simply by regulating the amount of enzyme, by regulating the gene expression from which that enzyme is produced. We're going to spend some time talking about inhibition. Inhibition is decreasing the rate at which a reaction occurs by inhibiting the activity of an enzyme. The inhibitors are compounds, usually small molecules, that inhibit or reduce the activity of a given enzyme. Now there are two different types of inhibitors that affect the enzyme kinetics in different ways. One is called a competitive inhibitor, and one is called a non-competitive inhibitor. Competitive inhibitors are small molecules that are similar in structure to the enzyme substrate, but they can't be converted to the product. And so they're able to sit in the active site of the enzyme and block access of the substrate to the enzyme, and that blocks the conversion of substrate to product. Now what happens when you do this is you change the apparent Km of the enzyme without changing the Vmax. Now the Vmax stays exactly the same. The maximal rate at which that enzyme can attain is still the same. But the concentration of the substrate that's required to make it half maximal is increased now because you have a small molecule competing with the substrate for the enzyme. And so the apparent Km of the enzyme will go up as you inhibit the enzyme with a competitive inhibitor. And this is shown graphically on the right. So in the regular curve, what you see is a normal rate of reaction shown at the top where there's no inhibitor, and then as you increase the amount of inhibitor, you see that the curve flattens a little bit, it decreases. It still reaches the same Vmax in the end, but the slope of that curve is decreased a little bit. Now if you try to map up the Km against that, what you'll find is that as you increase the amount of inhibitor, the substrate concentration at which that reaction is half maximal moves more and more to the right. That is, the Km increases. Now if you look at the Lineweaver-Burke plot, which is shown below that, you see a little bit different picture. The line at the bottom where there's no inhibitor. And you can see that the slope of that line is relatively flat. What happens when you increase the apparent Km is that the slope of that line increases. Remember that the slope of that line is defined by Km over Vmax. So as you increase the Km, the slope of that line is going to increase. But because the Vmax doesn't change, the y-intercept stays the same. So you can see that the line crosses the y-axis in exactly the same place, no matter how much inhibitor you add to the system. Now, non-competitive inhibitors are different. They interact with the enzyme at the other side, and they actually affect the ability of the enzyme to catalyze the reaction, and in doing so, they reduce the Vmax of the enzyme without affecting the Km. The Km is unaffected because it's not competing with the substrate for access to the enzyme. What it's actually doing is decreasing the ability of the enzyme to catalyze the reaction. And so Vmax goes down, Km stays the same. And that's reflected in the Michaelis-Menten plot, which is shown on the right top. What you see is when there's no inhibitor, you have a normal line. As you increase inhibitor, you can see that the Vmax, the point at which the curve flattens out, decreases with increasing inhibition. But the point at which each of those reactions is half maximal stays approximately the same. And you can see the same thing in the Lineweaver-Burke plot. In this case, because you're altering the Vmax, you're changing both the slope and the y-intercept of this line. Remember that the slope is defined as Km over Vmax. So as you decrease Vmax, the slope will increase. And so you can see as you increase the amount of inhibitor, the slope of each of these lines gets successively higher. Now, it also changes the y-intercept. Remember that y-intercept is defined as 1 over Vmax. So as you decrease the Vmax, the y-intercept is going to increase. And you can see that each of these lines has a sequentially increased y-intercept. Okay, so consider this question. This will test your understanding of what we just talked about. Which of the following will be unchanged in a Lineweaver-Burke plot of an enzyme with and without a non-competitive inhibitor? Is it the Vmax? Is it the slope? Is it the y-intercept, or sorry, the x-intercept, or the y-intercept? The answer is C, x-intercept. Now, we didn't talk about this before, but the x-intercept is defined as 1 minus 1 divided by the Km. Remember that when you have a non-competitive inhibitor, you reduce the Vmax without affecting the Km. So if you, in a non-competitive inhibitor, you're going to affect the slope, which is defined as Km over Vmax, and the y-intercept, which is defined as 1 over Vmax. The x-intercept depends only upon the Km, and because the Km doesn't change, then the x-intercept doesn't change. And that's illustrated nicely in the Lineweaver-Burke plots to the right. So here's a summary of how inhibitors work, and this is a really important thing for you to understand if you're going to understand inhibition of enzymes. So for a competitive inhibitor, you're going to increase the slope of the Lineweaver-Burke plot. The y-intercept will not change, and the x-intercept will actually increase. Remember, because you're increasing the Km. With a non-competitive inhibitor, you're decreasing the Vmax. Consequently, the slope will increase, the y-intercept will increase, but the x-intercept will stay the same. So that's an example of inhibition. Now, enzymes can be regulated in other ways as well. For example, you can have cooperative regulation. Some enzymes are comprised of multiple subunits with multiple active sites. And in many cases, these active sites interact with each other cooperatively. That is, when a substrate binds one subunit, it affects or facilitates the binding of another substrate molecule at another site. And so what happens then is, as the enzyme complex binds more molecules, it becomes increasingly effective at catalyzing the reaction. The rate of the reaction increases. And so what happens then is you get a sigmoid curve. That is, as you increase the substrate concentration, the rate of increase becomes faster and faster and faster until it reaches a point where it becomes saturated and it levels out again. And that gives you the sigmoid-shaped curve. Now, this is different than an enzyme which is not cooperative, which shows just the rectangular hyperbola like we showed before. Now, there's also what we call allosteric regulation. Allosteric interactions can occur between an enzyme and a non-substrate effector. These effectors bind at sites other than those bound by the substrate, and they either increase or decrease the enzyme activity. So a positive allosteric effector will increase the Vmax and cause an increase in the slope of the line as we show here. A negative allosteric effector will decrease the Vmax, and so it'll have a flatter slope, as shown with the red line in the diagram below. Here's some other forms of regulation that we'll talk about just briefly. Covalent modification. The most common modification is phosphorylation, the addition of a phosphate group to an enzyme, which can either increase or decrease its activity. And when we talk about metabolic processes, we'll describe cases of both of those, both where phosphorylation increases or decreases the activity of an enzyme. You can also affect it by protein-protein interactions. Proteins can serve as regulators by binding to and altering the enzyme function. Enzyme activity can also be regulated by increasing or decreasing the transcription, translation, or degradation of the enzymes. And in that, you're regulating enzyme activity by regulating the number of enzyme molecules in a cell. So this ends the section.